of the other things that you really played a big role in is understanding and your work in the BMAL1
Oh yeah, yeah. That's
function
my
in skeletal muscle mass, regulating sleep deprivation. Can you talk a little bit about that?
yeah. So this is actually why I was so excited to be on your podcast. You know, giving your very rich history and research that you've done and skeletal muscle physiology is so this is, I guess, my claim to fame in the sleep field, and what I'll, no matter what work I do from here on out, this is like what I'll be known for. So essentially and this wasn't just me. Like, I was a postdoc in the lab at the time, but I was the lead for the project. This is a collaboration between me and my postdoc mentor, Dr. Katema Paul at Morehouse School of Medicine, and then our collaborators at UT Southwestern, Joe Takahashi, and then Karen Esser, who's a skeletal muscle physiologist. She's now at University of Florida. So essentially, we know that every single tissue of the body possesses a self-sustaining biological clock. So you have the central biological clock in the hypothalamus of the brain, known as the suprachiasmatic nucleus. But then there are a series of peripheral clocks that have essentially been identified in every tissue of the body, to include even fat cells. And so Karen Esser, she's the one who discovered the biological clock in skeletal muscle and found, the same circadian clock gene factors that create the self-sustaining 24-hour rhythm. We took her model and essentially, through the help of Joe Takahashi at UT Southwestern, genetically engineered a mouse model where you could disrupt the expression of these clock genes in either the brain or the skeletal muscle.